I think we should start now. And hello, everyone. Thanks for joining us on Friday night. Welcome to our February academic seminar held by Boston Taiwanese Biotechnology Association, BTBA. And I'm Ming-Chieh, one of the directors of academia team at BTBA working with 𝗍. And this is our February academic seminar, and we will have our next academic seminar on March 11th, and also industry workshop at the last weekend of the March. And also in April, we will invite the senior editor from the Cell journal and to give us editorial workshop to tell us her story about being an editor and how to publish paper on Cell. And so let me welcome our moderator today, 为智夫医师. He is currently in the PhD program of Harvard T.H. Chan School of Public Health. Let's welcome 智夫.
Hello, everyone. Good evening. And it is my pleasure to see all of you here. And tonight, we'll have two great speakers, Dr. Chen Pei-Jun from UT Southwestern and Dr. Ming-Chieh from MIT. And before we start, we would like to ask everyone to turn on your camera and we'll have a group photo first. So please turn on your camera if it's okay for you, and we'll be taking a group photo together. Right, so we have many people here tonight, and we are going to take the photo. So please to get a camera, and 3, 2, 1. Okay, let's take another one. 3, 2, 1. Okay, so let's introduce our first presenter. On behalf of BTBA, it's our great pleasure and honor to invite Dr. Chen Pei-Jun, Chen, for our first talk of today's seminar. Dr. Chen received his PhD from Stanford University and joined the lab of Jonathan Weissman at UCSF, where he developed various sequencing and CRISPR-based screens to dissect the protein coding capacity of the human genome, specifically uncovering pervasive non-canonical translation and the whole universe of novel annotated proteins. In October of 2020, he joined the faculty at UT Southwestern, where he focused on the study on the regulatory roles of non-canonical translation and the annotated proteome in health and disease. Dr. Chen's topic today will be illuminating the dark matter of the proteome. Without further ado, let's welcome Dr. Chen. The floor is yours.
Well, thank you so much for the kind introduction. Let me share my screen first. All right, can everyone see my screen? Yes? No? Move this out.
Yeah, it looks good.
Can people see my screen? Yes?
Yeah.
Yes.
It works well.
Can people see my pointer? Yes, okay, cool. So I want to thank the PTBA for the invitation to share my work today. And thank you all for coming on a Friday night. So I'm an assistant professor at UT Southwestern Medical Center, and my lab is generally interested in genomics and gene regulation. And today I'll share with you 3 short stories. First on developing CRISPR-based tools, and second on developing CRISPR-based screens, and the third on using genomics and CRISPR screens to illuminate the dark matter of the genome. And hopefully you all can take home some new concepts today, not only on how fundamental assumptions in biology is actually much more complicated than what we think, but also really using a repertoire of enabling technologies to solve open questions. So as you all know, CRISPR-Cas9 consists of a Cas9 protein targeted to a specific location in the genome as specified by the sequence of a single guide RNA. So in a wild type nuclease version of Cas9, Cas9 will make a double-stranded break in the DNA. And the resulting double-stranded break will actually be repaired by endogenous repair mechanisms. But this repair is not perfect and will usually generate small indels or small insertions or deletions. And this mutation will result in a knockout of the target sequence and gene. And this is how we usually use CRISPR-Cas9 to generate knockouts. However, the problem is that DNA double-stranded breaks are often toxic to the cell. Furthermore, DNA repair is often unpredictable. And even though we hope for small indels, at times there may be large deletions, off-target mutations, and even chromosomal rearrangements. And this really limits its utility in therapeutics and in biology. And in biology, a clonal cell line needs to be generated, which can be quite tedious. However, another way Cas9 can be utilized is to create a so-called nuclease deactivated version, or dead Cas9, or just dCas9. And this provides a powerful tool to target dCas9 protein to specific locations in the genome without creating double-stranded breaks. For example, by fusing GFP to dCas9, it can be used to image genomic locations. And dCas9 can also be used to deliver regulators to specific sites in the genome. So for example, dCas9 can be fused to transcriptional activators and repressors. And this is called CRISPR-A and CRISPR-I to target to transcriptional start sites to turn on or off transcription. And for the case of CRISPR-I, dCas9 is fused to a protein domain called CRAB. And the CRAB domain actually recruits other factors to deposit methyl marks on the histone to silence transcription. So CRISPR-I and A can be used to fine-tune gene expression that does not create a double-stranded break as opposed to CRISPR cutting. However, the problem is that CRISPR-IA needs to be expressed constantly. And this really limits their utility in therapeutics because obviously you don't want to be continuously expressing an exogenous protein. And for biological applications, this means that a stable cell line will need to be generated. So to get around these problems, we aim to create a CRISPR fusion protein that can silence genes like CRISPR-I, but only need a transient expression, allowing for the permanent gene silencing without changing DNA sequence. So we created a novel fusion CRISPR protein consisting of the same dCas9, the CRAB domain in CRISPR-I for repressive histone modifications, but we added two other protein domains, as DNMT3A and DNMT3L, to methylate the DNA. And we call this fusion protein CRISPR-off. And this combination of histone marks and methylated DNA creates a so-called epigenetic memory. So this repressive marks, these repressive methyl marks are actually memorized and passed on from generation to generation. And we can see here that the transient expression of CRISPR-off with the guide RNA creates a long-term silencing of the target gene. And this is even when there's no longer expression of the CRISPR-off protein. And we actually tested this heritable silencing for up to a year, and the silencing still persists. On the other hand, for CRISPR-I, in which there isn't de novo DNA methylation, we can see that the silencing drops off after around 10 days. And to test for specificity, we did RNA-seq to look at global expression. Importantly, we see that the introduction of CRISPR-off machinery does not induce changes in the transcriptome, whereas knockdowns of genes are actually very, very specific. And by whole genome bisulfite sequencing, you can see that the methylation is also extremely specific. And we also created another fusion protein called CRISPR-ON. And CRISPR-ON can be used to revert the silencing and reactivate gene expression. And finally, we explored the therapeutic potential for CRISPR-off. So as one example application, we all know that iPSCs can be used to generate customized cells through gene editing that can then be used to differentiate into multiple cell types. However, CRISPR cutting changes the DNA sequence and induces DNA cutting, which can be toxic in stem cells. And so we wanted to test whether CRISPR-off can provide an alternative to gene editing in iPSCs, and wanted to test whether gene silencing can be sustained through differentiation. So here we transiently express CRISPR-off to silence our target gene, and we passage the iPSC for a month. And we can see that after a month, the gene is still silenced. Then we can induce differentiation of the iPSCs into neurons, and we can see that the gene silencing actually persists even through neuronal differentiation, as quantified here in red. And this is really interesting because somehow this epigenetic memory can be sustained even during differentiation, a process where there's global changes in the epigenetic profile. So CRISPR-off and CRISPR-on are yet another tool in this toolbox of CRISPR, and they can be used not only in therapeutics, but also in cellular reprogramming and engineering, as well as a tool to study epigenetics. So now I want to switch gears and expand our scope a little bit. So CRISPR is not only useful for targeted gene regulation or mutations, but CRISPR can also be adapted in a high throughput pooled CRISPR screen format. And this is very useful for the discovery of key genes or genetic sequences that elicit a specific function or phenotype. So here we can clone a library of guide RNAs and package them into lentiviral library and infect a pool of Cas9 expressing cells, such that each cell expresses a specific guide RNA targeting a specific gene. So in a pooled fashion, we can generate a genome-wide knockouts. And the cells with a gene knocked out as functional required for cell survival will have a growth defect. For example, here we can see that the orange cell have a lower count. And this suggests that the knockout is functional. We can then measure guide RNA abundance from deep sequencing as a proxy for cell count and calculate a growth phenotype score. So typically, these high throughput screens have been performed using cell lines, which is low cost and high throughput. But it is actually quite difficult to recapitulate disease models and progression with cell lines. On the other hand, mouse models and primate cell models can recapitulate in vivo conditions, are often low throughput and very expensive. So human organoids, which are essentially primary human cells that can be passaged long-term similar to a cell line, provide really a nice hybrid between a cell line and in vivo models. And using organoids, we are able to model disease and cancer progression, and in principle is amenable to genome scale genetic screens similar to cell lines. So first, we engineered the full suite of CRISPR machinery, CRISPR cutting, CRISPR-I and CRISPR-A into human gastric cancer organoids. And introducing SOX2 targeting sgRNA eliminates SOX2 expression for CRISPR cutting and CRISPR-I. And targeting CRISPR-A increases the expression of SOX2. So as a proof of principle, we performed a CRISPR screen on these organoids. However, in addition to monitoring growth defect, which is negative selection, we also added a positive selection, which is a drug treatment, where we introduced the drug cisplatin. And cisplatin is a common chemotherapy drug for gastric cancers. And it works by cross-linking with purine bases and resulting in DNA damage and cell death. So by comparing the drug-treated population and the no treatment population, this will allow us to determine drug sensitivity phenotype. And this this scatter plot plots the score, the phenotype score from our CRISPR-I and CRISPR-A screen, where a positive score is a growth defect and a negative score is a growth advantage. And we can see here essential genes for cancer growth. And notably, as expected, the CRISPR-A or overexpression of the MYC gene results in a growth advantage. And for the addition of the drug cisplatin, we can think of this drug sensitivity phenotype as a chemical genetic interaction. So the knockdown of a gene results in a particular fitness defect. And the addition of a drug will also result in a separate fitness defect. With the addition of both the drug and the gene, we can get three possibilities. If there's no chemical genetic interaction, we get the summation of the two fitness defect scores alone. On the other hand, for synthetic lethal interactions, we get a much stronger defect than from both alone. And this has large implications for chemotherapy since it provides a roadmap to using combinatorial drug treatments. If we look at And when you look at the top, the hits in our screen, we can see that the top interactors are actually DNA repair genes. And since the mechanism of cisplatin is to result in DNA damage, it makes sense that knocking down the DNA repair genes will result in a much stronger and severe phenotype. And as you can imagine, if we have another drug that blocks one of these proteins, it might enhance the effect of cisplatin in killing cancer cells. Now we can actually go one step deeper. So instead of monitoring cellular growth as a phenotype, we can use perturb-seq to measure simultaneously the single cell RNA-seq profiles as well as the guide RNA identity in each cell. So in a pooled fashion, what we can get is we can knock out a specific ORF and get how the transcriptome changes upon that knockout. So this technique called perturb-seq allows us to go beyond the coarse phenotype of CRISPR screens where we can only identify hits with growth phenotypes. And perturb-seq provides an extremely rich data set for decomposing cell identity, gene set enrichment analysis, and importantly, provides a very high dimensional phenotypic readout that is much more complex than just a phenotypic score. So for example, we can see that the addition of the drug gives us a transcriptional phenotype as represented in this heat map here. If we knock down a gene with no drug, in this case, the gene 𝗦𝗃𝗃𝗃6, which is one of our top hits from our CRISPR screen, we get another transcriptional phenotype profile. And if there is no interaction between a drug and a gene, then the theoretical summation of the two transcriptional phenotypes should give you something like this. However, that's not what we saw. Instead, we get a drastically different and a stronger transcriptional phenotype. And here we can see that perturb-seq provides really a high dimensional readout for drug synthetic lethality. In particular, we observe that there's one gene that is significantly synergistically repressed, and this gene is GMDS. So GMDS is actually one of the key enzymes in the fucosylation pathway. And if we only focus on GMDS, we can see that this synergistic repression is not only for 𝗦𝗃𝗃𝗃6, but also for 𝗦𝗃𝗃𝗃4, 𝗦𝗃𝗃8, as well as the knockdown of other genes involved in nucleotide excision repair. So we're still working on the mechanisms, but really the results indicate a novel crosstalk between fucosylation and cisplatin sensitivity. And more broadly, between glycosylation and DNA repair. And this really demonstrates the power of CRISPR screens and perturb-seq in organoids to map out unknown pathways and for discovering new biology. So now I want to switch gears once again to talk about how CRISPR screens and genomics can be used to define the full protein capacity of the human genome. So traditionally, people usually define a gene as a term interchangeable with the terms protein or transcript. And in fact, in my previous two short stories, I have made that assumption. So people usually will target CRISPR-I to silence transcription of the entire transcript, or target CRISPR cutting to the coding sequence of the transcript to generate a knockout. And people usually assume that these two are equivalent, but this is actually based on this monocistronic assumption, or one gene, one protein hypothesis. But is this really true? So the answer is no. We typically think of messenger RNAs as messengers to produce proteins. So we usually tend to focus on the protein coding sequence, or the main ORF, and tend to ignore the untranslated region in the 5' UTR and the 3' UTR. However, there may be alternative start codons encoding for non-canonical open reading frames that are located either upstream in the 5' untranslated region called U-ORFs or upstream ORFs, in the 3' UTR called downstream ORFs, or even overlapping the main coding sequence in a different reading frame. And these non-canonical ORFs are short and encode putative small proteins called microproteins. For example, PTEN, a very well-known tumor suppressor gene, actually encodes a 33 amino acid upstream microprotein. And 𝗠𝗨, a gene with important implications in various diseases, actually encodes a 170 amino acid protein that is entirely nested within an annotated coding sequence in a different reading frame. And these alternative start codons can actually encode variants of annotated proteins, either extensions or truncations. So for example, this MYC oncogene can encode an extended variant translated by a near cognate CUG start codon. So there is really a whole universe of these novel proteins that were previously basically hidden in plain sight. So even for well-studied genes like MYC and PTEN. And so this is very clear that the definition of the gene is much more complicated than what we think. And even for non-coding RNAs, such as link RNAs, there's now increasing examples of these short open reading frames on link RNAs encoding microproteins. And this term link RNA, because of this, has become somewhat of a misnomer. So all of these microproteins have been gaining a lot of interest in the past few years, with many examples being discovered and being shown to perform important functions in a wide range of biological processes, ranging from immune response, metabolism, development, to cancer. So this problem arises mainly because annotating human protein coding regions relied on several stringent and rather arbitrary criteria to avoid false positive annotations, such as the use of a 100 amino acids size cutoff, a monocistronic one gene, one protein hypothesis, an AUG start codon, and high sequence conservations. And these rules have been necessary and undoubtedly effective in mapping the genome. However, the very stringency that makes them so valuable also limits our ability to discover non-conventional gene architectures. So the question now is, are these examples just isolated ones, or is this architecture widespread? So emerging data, including our own, suggest yes, and my lab sort of aims to characterize the so-called full protein coding capacity of the human genome. So here, to answer this, we developed a tiered strategy to globally discover and functionally characterize this unannotated proteome. So to discover novel ORFs, we can directly profile these non-canonical translation events globally by a technique called ribosome profiling, or RiboSeq, where we RNase treat the ribosome-associated RNA to get ribosome-protected footprints. And purification and library preparation gives us short fragments of RNA that can then be sequenced and aligned back to the genome to give us ribosome densities. And this provides us with a genome-wide view of ribosome translation. From this, we can then computationally identify novel open reading frames that were previously unannotated, taking advantage of signatures of ribosome densities to call high confidence ORFs. So ribosome profiling can give us translated ORFs, but to screen for functional ORFs, we can then take advantage of CRISPR-Cas9 to target Cas9 to the novel ORFs identified from our ribosome profiling screen. So this genome-wide CRISPR ORF screen will then comprehensively determine functional ORFs encoding putative microproteins. And finally, the candidate microproteins can be mechanistically characterized using a repertoire of in vitro and in vivo assays, such as mass spec, microscopy, and structural techniques. So from this, we identified, in addition to around 10,000 annotated coding regions, really thousands of variants of annotated proteins, such as truncation, extensions, and different isoforms, as well as distinct open reading frames on non-coding RNAs, upstream, downstream, and even internal ORFs that may encode putative microproteins. In addition to the canonical AUG start codon, there's initiation at near cognate CUG, GUG, and even UUG start codons. And importantly, we have found that these novel ORFs are often upregulated during many biological processes, such as tumor genesis and stress. And this implicates their important role in these various disease states. So I showed that link RNAs, at least some link RNAs, encode microproteins and play diverse roles. And here, there's 3 examples of novel link RNA microproteins, which are 62, 59, and 70 amino acids, respectively, localized to the mitochondria, ER, and plasma membrane. So IP mass spec results shown in these volcano plots show interactions consistent with their localizations. For example, for this mitochondrial localized peptide, it interacts with many mitochondria-related proteins, such as the cytochrome C oxidase complex. Whereas this ER localized peptide interacts with a lot of proteins involved with trafficking, and this plasma membrane localized peptide interacts with a lot of protein channels and cell surface proteins. So the combined localization and physical interactions give us a very good clue on what the peptide is doing. and this provides a blueprint for future in-depth studies. So link RNAs, at least some link RNAs, encode microproteins and play diverse roles in cells. And now we turn our attention to upstream ORFs or UORFs, and also show that the UORFs that are encoded in the 5' UTR of canonical protein, normally thought to be translational modulators, can also encode microproteins. And the UORF encoded protein is actually distinct from the downstream encoded protein on the same mRNA. And interestingly, I also found cases where this UORF peptide actually colocalized and formed a stable physical complex with the downstream encoded protein. So for example, for this MIF1 gene, the co-IP of the MIF1 UORF peptide shows that it interacts with the downstream MIF1 canonical protein. And the peptide localizes to the mitochondria, also consistent with the localization of the canonical MIF1 protein, further supporting the interaction between the UORF peptide and the main protein. And the canonical MIF1 protein regulates mitochondrial fission and fusion. So I speculate that this UORF peptide may have similar roles. So we explored its function on mitochondrial morphology. So in wild-type cells, the normal morphology of mitochondria sort of looks like this. We observe that the overexpression of the MIF1 UORF peptide alone induces a fragmented mitochondrial phenotype, which is increased fission, whereas the knockout of the MIF1 UORF resulted in a tubular and more elongated mitochondrial phenotype, which is increased fusion. So this physical interaction between the two proteins on the same transcript is particularly interesting. So since this implies an additional layer of regulation beyond the modulatory nature of UORFs. And the principle underlying these type of polycistronic and bicistronic architecture will require further mechanistic studies. But I demonstrate that the use of these polycistronic architecture in human, a strategy commonly used and recognized only in bacteria and viruses to co-express functionally related proteins. So just to sum up, there really is a whole universe of these dark matter of the proteome. And in my lab, the goal is to really study the functional roles in these novel proteins and translational control in the context of cellular and physiological transformations, such as tumorigenesis and stress. And finally, we were also very keen on developing novel technologies to specifically perturb different systems as well as measure complex phenotypes. So I'll be moving my lab to the Bay Area in California to join Altos Labs. For those who may not be familiar with Altos, Altos is a new startup/research institute that is focused on basic science research in cellular aging, as well as reprogramming and protein homeostasis. And so they recruited some of the best scientists around the world. And the goal is to create a really highly collaborative and academic-like environment focused on basic research. So if you know someone that might be interested in genomics, functional genomics, tech development, CRISPR, gene regulation, and or microproteins, please contact me. So I'd like to thank you for your attention. I would also like to thank my lab members and also my postdoc mentor, Jonathan Wiseman, members of the Wiseman Lab, as well as collaborators. And finally, I would like to thank my past and present funding sources, Jane Coffin-Jowd's postdoc fellowship, an endowed scholars award from UT Southwestern, a faculty award, and the NIH K99 Pathway to Independence Award. Thank you, everyone.
Thank you so much, Dr. Chen. It's a great presentation, and I learned a lot. So any of the questions from the audience, please feel free to unmute yourself for questions. As the questions are pouring in, maybe I can grab this chance to ask about Dr. Chen, ask a very preliminary, naive question for Dr. Chen. Like, would you mind provide like one or two potential application that can be translated in clinical settings? I think the gene-drug interactions are really interesting, and it would be great if you can talk more about like 1 to 2 more examples, like all this technique, like all the discovery of dark matter can lead us to the next era.
Yeah, so of course, so obviously various CRISPR techniques are used by a lot of startups. to try to cure a lot of diseases. So if you're interested, CRISPR-off is getting a spin-off to a startup called Chromo Medicine that's located in Boston. And also, for these novel microproteins, I think there's a lot of potential because they are sort of novel targets, right? So if you can find novel microproteins that are specific to certain cancer, then these are druggable targets. And these microproteins are also small, right? So they themselves can actually be used as modulators of other disease-relevant proteins. So it can also be used as a gene therapy approach. So there is also a spin-off of that called 𝗦𝗲𝗲𝗿 Therapeutics that we recently started based in San Diego. So if anyone is interested in, please feel free to contact me.
Wow, that's really cool. And I think, are there any questions from the audience? Please feel free to unmute yourself. Yang-Xin?
Hey, Jing, it's really nice talk. I have a question regarding to the CRISPR-off. It utilized three component, CRAB, DNA3A, and DNA3L.
Mm-hmm.
Are those essential? If you take out one of them, are they still functional? Because it seems like it will be hard to bring three components together and also take Cas9 together to any kind of cell like primary cell. So
Yes.
I think it will be essential to know whether we can bring less to the cell.
Yeah, that's a good question. So these are created as a fusion protein, right? So it's one entity only. But the problem is, as you alluded to, it is one large fusion protein. So it's not amenable to a lot of like AAV delivery techniques. So regarding whether we can shorten it, so obviously there are ways in which we're working towards right now. For example, using a smaller CRISPR protein, or but right now in our hands, those three proteins are essential for these long-term sustained memory silencing. Yeah.
Okay. Yeah. Thank you so much.
Nice. Shih, go ahead.
Hi, Jay, nice to talk. So I have other questions. So I know some other lab, they are developing other systems to maybe compare to CRISPR-Cas9. I was wondering whether you have heard from another group called Reproductive DNA to do precise genomic editing. Yeah, and what's the difference between that and the Cas9 system to, for example, to target the heterochromatin or the 𝗠tože/𝗿, the Cas9 targeting site? Thank you.
I'm actually not familiar with that. Yeah.
Oh, okay. So yeah, so my question is that, so because Cas9, there are some concern about maybe of targeting or 𝗠tože/𝗿. I was wondering whether the team also try to develop a system to overcome that problem to make Cas9 more efficient in the future or not?
No. Yeah. That's so far no, not yet. Yeah. But the good thing about CRISPR-I, CRISPR-A, or CRISPR-off is that they don't generate double-stranded breaks, right? So the off-target, even if there's off-target, I think the effects will be minimal as compared to just Cas9 cutting.
So thanks a lot for Dr. Chen's great presentation and all the great questions from the audience. And I really appreciate that. And let's turn to our second speaker, Dr. Ming-Chieh. Ming-Chieh graduated from National Taiwan University. He received his medical degree and PhD in biostatistics there. And his study mainly focused on the evaluation of network meta-analysis using structural model equation modeling. And he's currently a postdoctoral fellow in Clinical Machine Learning Group, Institute of Medical Engineering and Science in MIT. Dr. Shi's research focus on methodologies and applied analysis on healthcare data. And his topic today will be challenges in statistical methodologies for meta research. Without further ado, let's welcome Dr. Chih. The floor is yours.
Please wait a moment, I'll have to share my screen.
Take your time.
Okay. Can everyone see my screen now?
It's not in full screen mode. Maybe you can put it in.
Okay.
It looks great.
Okay. So I think without further ado, I'll start now. So thank you for the opportunity for me to share some of my previous work and my current work on statistical methodologies, predominantly on causal inference. So I'm Ming-Chieh, and as Dr. Wei had said, I graduated from National Taiwan University with an MD degree, and then I pursued a career in biostatistics. And during my doctoral years, I actually did some research on the methodologies of network meta-analysis, which I will elaborate later. And currently, I'm in the clinical machine learning lab in MIT. So basically, we're doing machine learning methods on large health claims data, and also some causal inference stuff. So the content of today's talk is about meta research. So meta research is actually a rising field that is researching other researchers. So hence the name meta research. So in meta research, we want to we can divide it into 5 different categories, methods, reporting, evaluation, reproducibility, and incentives. So basically, we want methods that are adequate, that are solid, and can answer the questions that we're invested in. And we want a transparent and comprehensive reporting. And we want to evaluate the results in the robust way. And also, we want our studies to be reproducible so that people can reproduce our studies. And for that, we want to have incentives for researchers to publish high quality studies with sound methodologies. And within these 5 dimensions, there is evaluation. So meta-analysis is actually within this dimension. So given that we have a lot of different studies that are studying on the same topic, we want to know that what is the aggregate evidence, or we want to summarize the evidence for the same quantities of interest that is reported by different studies. And actually, meta-analysis is not new. It goes way back from 1904. You can see that this is a previous paper by Carl Pearson. The Carl Pearson is just the Pearson proposed the Pearson correlation. You can see that there are different studies that has reported the correlation between mortality and inoculation for enteric fever. So basically, these studies report different results, and even some studies reported a negative correlation. And what Pearson did is he tried to, given these 6 numbers, he tried to use a statistical method to use a robust way to combine these numbers into the mean value of 0.193. So basically, this is a nutshell of what meta-analysis do. So it is a statistical method, and we try to aggregate and summarize evidence using solid statistical theories. Okay, so that we know that we can combine the studies, but why do we want to combine the studies? In the circumstance where there is a large a randomized clinical trial on this subject, then why should we combine this large clinical trial with little clinical trials to give a summarize of evidence? Why can't we just look at the result for the large clinical trial? There are a lot of reasons why we need meta-analysis. The first one is maybe we want to reduce variation. In the case where all this all the studies are low in power, then maybe we want to like reduce the imprecision of our estimates. And second, maybe like different studies have conflicting evidence, like for the comparison between two different drugs. Like this study says that this drug is effective, and that study says that the drug is actually harmful. So we want to use a statistical method to like resolve the conflicts of between these evidence. And also because like studies can be underpowered, and underpowered studies can give you inconclusive conclusions, or like maybe you can produce spurious results because like your study is underpowered. So meta-analyses can attain power by combining these studies and give us a more robust conclusion. And also we can account for heterogeneity in meta-analysis. To know what is heterogeneity, we can look at the following plot. Sorry. So like basically what ideally we would think that every study is estimating the same quantity, like the effect between the effect difference between two drugs. So like for study 1, study 2, study 3, they should be estimating the same like risk ratio or risk difference. But actually, because like different studies recruit different people, so if there is like a heterogeneity in population, there will be heterogeneity in study effects. So for instance, maybe like this study is conducted in Asia. Then like for Asians, maybe the treatment effect is lower, but for like Europeans, the treatment effect is higher. So like the true treatment effect for each study is actually not the same, and they follow a distribution. So with meta-analysis, you do not look at only one study and assume that it is estimating the true like grand mean of the treatment effect. You actually like look at different studies and average between these studies to get a grand mean, like this is like true treatment effect. And last, we can explore effect modifiers using meta-analysis. So like we said that there may be heterogeneity that we can detect, but if we have further information on the like distribution of effect modifiers like gender or like ethnicity, then we can further know that if any like ethnicity is related to like the treatment effect. Okay, so that is like a overview of meta-analysis and what meta-analysis do. But actually, like when you start to use meta-analysis, then what the statistical method you use actually depends on what you want to, what you have at hand, like what you want to know and what studies have you collected. For instance, like based on the nature of quantity of interest, we can have like different types of meta-analysis. Like conventional meta-analysis, like deal with like for instance, like the effect difference of different drugs, and like there are a bunch of studies that studied on this quantity, and we want to like combine them. Or like maybe you want to do dose response meta-analysis. What this means is that like more often, like in nutritional study, you want to know that like the intake of a certain kind of nutrition, like the amount of intake should be related to the effect of the like intake of nutrition to your outcome. So actually, like different studies may have like different doses of this intake, and you can actually plot a curve based on different results on different studies. And also, like there may be some diagnosis tests that you want to know that what is sensitivity or specificity. Like currently, like we have like a lot of different diagnosis tests for like COVID-19. So you want to know that what's the actual sensitivity and specificity, like based on the reported results from like different companies. So you can use meta-analysis to address the difference of the reported results. And fourth, we can, if you want to compare different, like more than 2 different drugs, and we want to compare like 3 or more drugs and know their like effect difference, then you can use network meta-analysis, which we will like talk more about later. Okay, and also like based on the data availability, like previously, traditionally, we only have aggregate data. That means that you only have the estimate of the treatment effect and like the 95% confidence interval. So you have aggregate data and you do not have any individual patient data. But now like more and more studies are providing the individual patient data, and we can do like a better analysis using individual patient data. And last but not least, there is a new methodology called trial sequential analysis, which lets to assess if there is enough data for this kind of topic, given that there are like multiple studies have been done, and you want to know that if these studies is sufficient to give you a solid conclusion on like these two drugs are actually different or like actually not different. Okay, let's talk about like why do we want to extend from like traditional meta-analysis to network meta-analysis. This is because like most diseases may have like multiple treatments available, and it may not be feasible to conduct trials like it includes all available treatments. This is especially true for like cancer drugs, because like different companies would not do like a head-to-head trials between their like new drugs. They will compare their new drugs to a placebo. So actually, like if you want to compare like the relative efficacy between two new drugs, then actually you do not have this randomized trial. So you can use network meta-analysis to use the information of A versus B and A versus C to get the information on B versus C. And also sometimes you actually have direct comparison, but you would like to combine the direct evidence between like a B versus C and the indirect evidence of B versus A plus A versus C. So network meta-analysis, assuming that the network is coherent, it can give you a more robust conclusion. As an example, we can look at this like Ming-Chieh paper. It is like comparing the efficacy and safety of some surgical treatments for benign prostatic hyperplasia. And you can see that actually there are a lot of different treatments, and some of the treatments do not have a direct comparison between them. For example, like for like monopolar TERP and the diode LEP, there are actually no randomized clinical trials directly comparing them. But using network meta-analysis, we can use the reported results for other comparisons to get indirect evidence between these two, so that you can compare. Like there is no monopolar TERP here because like it is used as a reference treatment. So like these treatments can actually like be compared to the like the reference treatment, which is monopolar TERP, using the network meta-analysis. But there are some cardinal assumptions on network meta-analysis. And conceptually, it's the transcriptivity. So we said about like if you want to know the difference between B and C, you just add the difference between B and A and like the difference between A and C. This is like implying that there is transitivity between like these two comparisons. So qualitatively, like these two studies should be similar in the sense that like the placebo they used should be like similar, or that the outcome assessment methods should be similar, like these follow-up periods should be similar. And also like a distribution of effect modifiers should be similar. So like it would be less ideal if like this study only included female and this study only included male. Then you're not comparing apple to apples and you're comparing apple to oranges. And for the data manifestation of the similarity, there is a notion of consistency. This means that like indirect evidence should always agree with direct evidence. For example, like for the comparison between B and C, the difference between B and C should be like the difference of A and C minus the difference of A and B. But actually, like the detection of possible inconsistency in network meta-analysis is crucial, but it is very challenging. Like if the consistency is violated, meaning that there's inconsistency, then actually like the whole result of network meta-analysis is not robust. But currently we have very limited methodologies on the detection of possible inconsistency. And there are two views of inconsistency based on the definition we have set. The first is like more intuitive, is the direct indirect evidence inconsistency, which is what we've said. So like a BC should be equal to BA and AC, like combined together. And another is the loop inconsistency, which means that like the evidence should be consistent within any loop. And currently we are using we're more using like the direct indirect evidence inconsistency because it is like more intuitive to use. So current models of detection of direct and indirect evidence inconsistencies involves like splitting the studies into two different groups. And we assume that these two groups are estimating different parameters, and then we compare the parameters between them. This is basically what current models are using. And these models can be fit under the like structural equational modeling network meta-analysis framework I developed back in my doctoral days. And the reason of why we want to use like structural equational modeling to like a model the network meta-analysis is because like it provides a unifying framework for several different types of models using network meta-analysis, and it can provide a immediate relaxation of strong model assumptions compared to like other frameworks. You cannot relax them. And also like for future use, like inconsistency models and like fit indices are easy to like assess in structural equational modeling. So actually like the main contribution of mine is that I analyze the evidence structure of network meta-analysis. Like the main theme is that like there may be some multi-arm studies. The definition of multi-arm studies is that studies that included 3 or more different treatments. So like for instance, you want to know like the difference between drug A and drug B. So there will be studies that only compare the drug A and drug B, which are like the direct pairwise studies. And there will be studies that included drug A, drug B, and others. There are multi-arm studies. So they also, but these studies also provide direct evidence, but they also provide other information other than the direct evidence of A versus B. And other studies are like those studies that did not simultaneously include A and B. And what I argued is that we have to be very meticulous in like dissecting the evidence provided by direct multi-arm studies, not like previous model which they just assume that different parameters are used in different studies. So we propose the evidence splitting model by requiring informational independence between direct and indirect evidence. And this is what the like a structural equational model would look like. Like this may seem like a little bit complex, but actually it is pretty straightforward to implement in like any standard SCM softwares. Okay, so we want to know that if our like new model on the like direct and indirect evidence inconsistency do better than previous previous models. So there are two different like criteria. So we want to there are two different qualities that we want the model to have. Like first is that changes in data constituting the indirect evidence should not affect the direct evidence estimate. Like this is very intuitive. Like if you're changing like the reported estimate of an indirect evidence, then it should not affect the direct evidence estimate you're getting. And second is that if the model implies that there is no indirect evidence, then the network meta-analysis estimate should be identical to the direct evidence estimate. And here we have like synthetic data where like we have a set of studies that represent a network meta-analysis, and we just play with like different a reported estimate of different studies. So let's assume that we're interested in like comparing A to B. Then actually, like if you modify the reported estimate for a study reporting A and C, then actually you should not have different estimates, different estimates for like different direct estimates for A and B, because you're just modifying A and C, right? So like in scenario 2 and 3, we're just modifying like the AC study and the BC study. But you can see that current method gives you different direct evidence estimates under these circumstances. So this is not ideal. And for scenario 4, you can see that although like in the pairwise meta-analysis, like the A and B, for A and B, the pairwise meta-analysis estimate is 2, and the network meta-analysis estimate is 3. That means that there is actually some kind of indirect evidence put into this meta-analysis. But you can see that this model shows that there is no indirect evidence. So this is also not ideal. And our evidence splitting model is doing like doing well in like every circumstances. Okay, so this is part of my like previous work, and I will talk a little bit about like my current work. So my current work in MIT involves like a causal inference in like medical data. So like one of the things I'm collaborating with is to discover the issues in meta-analysis of observational studies. So sometimes like the only evidence for a population of interest is from observational studies. Like for instance, like if you want to know like the treatment effect of a cancer drug, but you only have evidence from those who are stage 1 and 2 in RCTs, but you do not have evidence for those are stage 3 and 4 in RCTs. And more often than not, you will have like a larger database for like observational studies. So like for stage 3 and 4, for this subpopulation, you only have the observational study evidence. And observational studies have larger sample size, and they can provide more precise estimates. And we would like to like use these estimates, but they are also at large risk of bias compared to RCTs. So the usual strategy is to use a lot of different tools to assess if these observational studies have like a risk of bias. And then we just cross our fingers and hope for the best if we do not detect any possible bias. But we found that in the case where like other populations, like for instance, for our previous sample, like for the stage 1 and stage 2, like cancer patients, if there is evidence from both RCTs and observational studies, we can try to falsify possibly biased observational studies with RCTs and then produce conservative confidence intervals for the outcome of interest at the like non-overlapped population, which is stage 3 and 4 patients. So I can give you like a sneak peek at the results. So basically, this is for the meta-analysis. So like assuming that there are some bias studies in the observational side, if you do not do the falsification, then meta-analysis will actually give you like under coverage. Here, like you can see that this is this coverage probability. So we want the coverage probability to be around an 0.95. But you can see that meta-analysis have like a not good coverage. And the methods we are proposing, which are the blue one and the purple one, are actually performing well, especially for the blue one, the 𝗠XPCS. And this is like for another scenario where like the number of bias observational study varies. And you can see that like the 𝗠XPCS is actually have very good coverage probability. So we can get a very robust confidence intervals if we use the RCTs to falsify the studies. So for a brief summary, sophisticated meta-analytic techniques enables better utilization of available evidence, such as like network meta-analysis, but they are with the cost of additional assumptions, such as consistency. And we found that like the current evidence splitting model, we constructed under information independence, and it provides a correct evidence split, which is crucial to the inconsistency evaluation of network meta-analyses. And last, we found like a meta-analysis on observational studies can be made more robust to bias with external out-of-population information from RCTs. Okay, thank you for your listening, and I'm open to questions.
Thank you so much, Ming-Chieh. That's superb. And given the time limit, I welcome the audience for 2 to 3 questions before we end this talk. And Ming-Chieh, superb talk as always. I always enjoy listening from your presentations. I always learn a lot. So are there any questions from the audience? Please feel free to unmute yourself. If not, maybe I take the advantage as a moderator to ask Ming-Chieh brief, naive questions, if I may. So, yeah, Ming-Chieh, so I think it's really interesting to see how you meta-analyze those observational study with the help of RCTs. But may I ask one question, like for some of the exposure, for example, we do exposure site environmental AP. So there are exposures that will not have available RCTs. For example, like air pollution. We are not going to design an RCT for air pollution as a treatment arm. So in this scenario, what would be your suggestion to make it more robust in a real world setting?
Oh, so you're thinking that maybe there are not any evidence of RCT, even like
when there's
any subpopulations.
Yeah, when there's only observational, what will be your solution?
Yeah, like actually there are other things you can look, like for observational study, there are other techniques, like you can have like synthetic outcomes, like synthetic negative control outcomes. So basically the most the thing that we're worried about is like something like confounding bias or selection bias, right? So like basically we can try to find some endpoints that should not have difference between like exposed and non-exposed once you have controlled all the confounding bias or the selection bias. So like this is just like this does not, if like in the end like you analyze your study like using the negative control outcome and you see that there is actually no difference between like exposed and non-exposed, then this does not necessarily prove that there is no selection bias or confounding bias, but it's like a safety net for you to see that given that a bunch of different negative control outcomes, you do not see any evidence of like there is residual confounding bias or like other bias. Or also like you can construct positive outcomes. So like both negative and positive, like these synthetic outcomes serves as like a fail-safe for your like study design. Yeah.
That is great. Thanks a lot, Ming-Chieh. And are there any questions from the audience? Please feel free to unmute yourself for the questions. All right, count 1, 2, 3. So if there's no further questions from the audience, let me thank Dr. Shih and Dr. Chen again for their brilliant talk. We really enjoy having you in the Friday evening in Boston and Saturday morning in Taiwan. It's superb. And thank you all for all the audience participation today. And we'll have a monthly meeting following this seminar. Please join us with the link posted in the group chat. All right, so that's the end of today's seminar. Thank you again and wish you all the best.